Welcome everyone to this week's edition of the Extracellular Vesicle Club. I'm very happy to have two groups that are featured today. One of them is Eduardo Reategui's group at Ohio State University. And then we're also going to be hearing from Alex Johnson-Buck, who's with the Nils Walter group. And some of the other members of that group are going to be here today as well. So our topic today is single particle analysis and also single molecule analysis. There's been a lot of work that's been done in this area recently, a lot of really interesting papers that are coming out, but still plenty of ways and opportunities to push the envelope of what we can really detect and apply this as well to human health. I just want to make a few comments at the beginning here. My name is Ken Witwer. I'm the president of the International Society for Extracellular Vesicles, and I'm actually coming to you today from Germany. I was just in France at the French Society for Extracellular Vesicles meeting. It's a very exciting time in Strasbourg. We had some fabulous poster sessions, some really good oral sessions, and a lot of opportunities to talk about EV science. And there's some very good news. We had the final day of our submissions for the ICEF 2025 meeting in Vienna, Austria next April. And we had an increase in abstract submissions this year that was not 10%, not 20%, but actually about 50% greater than any previous year. So if you have not submitted an abstract to ICEF, please think about doing that at the late breaking period, because this is going to be a blockbuster meeting. It's going to be the biggest meeting that extracellular vesicles have ever seen before. So let's go ahead and get started with today's presentations. We're going to be hearing first from Eduardo and then from Alex. And I'd just like to remind everyone to please put your comments and questions in the chat box. I will then allow you to unmute yourselves to show your video if you want to at the end of the presentations, and just specify in your question whether it is for Alex or for Eduardo and their teams. So Eduardo, welcome. I know that you have some teaching duties today, and I really appreciate your joining us anyway, and being willing to give this presentation on a recent paper that your group has had. So thanks so much, and please go ahead. The screen sharing is yours.
Thank you very much, Ken, for the invitation. And yeah, so today I think I'm going to be describing about a recent paper in terms of characterization of viruses at the single particle level. And basically, I just want to start with how this idea started for us. So as an inspiration, we came across this paper in which basically it was described that there's many similarities between EVs and viruses. Okay, so some of them are highlighted here. So for example, we can be talking about that they have similar sizes. They also have similar type of cargo in terms of surface proteins, in terms of like nucleic acid content. And more importantly, also, we see that cells that are generated or infected cells that has a particular virus, it can also, their EVs can carry certain viral proteins and RNAs. I think that was actually very intriguing for us, and we started to explore this idea such that at that time also was the pandemic basically started, and it was sort of like a good match for us in terms of research direction. So here, for example, we have some initial characterization that we did in terms of like size range that we can have for EVs. This is a particular size range for EVs in which we can compare in terms of the SARS-CoV-2 virus, for example. Then we also start looking in terms of like if we can characterize these single particles as we did it before with EVs, since previously we actually developed a platform that allowed us to characterize proteins and RNAs in single EVs. So, and then we did this simple comparison in terms of, for example, if we can measure the fluorescent signal that came from S proteins from the SARS-CoV-2 virus, or for example, if we can measure another characteristic protein for general EV, like in this case from H1568 cells, like a CD63. So this was a good indication for us that actually we maybe can tune some of the platforms that we have developed before in terms of characterizing of certain proteins and RNAs that came from that are present in viruses. So What I'm going to be mainly talking today is basically the repurposing of a technology that we developed in my lab that was also published last year in Journal of Extracellular Vesicles, in which we demonstrate that actually when we immobilize EVs in a surface, actually it's possible to characterize, the surface content in terms of the different proteins that they have, and also different RNAs that may be inside. So the principle here basically is, as I'm going to be showing you here, this is sort of like a schematic of the device, which is divided in different wells, in which we will put different types of biofluid. In this case, for SARS-CoV-2, we primarily work with saliva and nasal swabs. And what we have in the surface is basically antibodies that would be targeting a particular subpopulation of the particles that we can have in the biofluid, like, for example, the SARS-CoV-2 particle and also the EVs. What's interesting about our method is that we actually can combine the readouts of all these different markers that we can have. For example, in terms of the virus particle, we can directly get measurements from the S protein from single viruses, and then we can also target with molecular beacons. We can target the inside of this particle and look for characteristics or basically target the RNA molecule that we have it here. For EVs that we have in the same sample, initially we find out that like the signal that we can have in terms of S protein, either way it's at the surface or inside, it was much more lower compared to the virus as we were expected. But for our surprise, we found that, for example, in terms of the viral RNA content that they have is actually pretty good. So it's actually, in terms of developing an assay that would allow us to do have a very high sensitivity, actually is very important for us. So the key features of our assay is basically what we are trying to do is the analysis in these individual particles. So basically, we are not lysing these particles. So we are not looking in principle to do any sort of like amplification step. We can target multiple regions of the RNA. Like in this case, for example, if we are interested in looking at some mutations that may be present in the RNA. And here in this table, we can have, for example, a number of probes that we use for targeting different regions of the RNA, including some mutations. So here's a characteristic readout that we have. So our readout is based on fluorescence. For this, we use total internal reflection fluorescent microscopy. And here, for example, we have individual particles in which we can see the S protein in fluorescence. Then if we target the RNA inside these particles, we can actually get a signal from them. And then also, if we look in the EVs, if we look at, we just change the color here to see the difference, we can actually see that we can have lots of this actually viral RNA that these EVs carry. So we can characterize this in terms of this histogram. And then, we can measure, for example, compare with samples that are not in samples in which those viral signatures are not present, and then, try to design an assay based on that. So here is just some images of individual particles. So this basically, what we are seeing here is an average of the signal per particle that we're having seen. So this is for example, these three images here are in terms of like perf microscopy. And here we can also have demonstrated that we can also measure this with STORM, which actually is for single molecule measurements. But in terms of like our main goal for this was the developing an assay, for actually high throughput characterization of a large number of patient samples. So we prefer to go with the average measurements per particle actually here, like in this case with her. Here are some analytical measurements, like in terms of sensitivity and specificity for the antigen measurements and also for the nucleic acid measurements. So this is something that we did a number of times, and we compared with the gold standard here, which was actually QRT-PCR, and we determined that our limit of detection is in the order of 2 to 100, much better than this gold standard measurements. So this is basically how it would look like in one of the images that we have, and then we will compile these histograms, and then we can just have also our average measurements based on a threshold that we have here. For us, it's very important to determine this threshold, because this is the one that we're going to use when we are trying to determine our cutoff values for our different assay that we have. So we can also expand this in terms of the detection of, for example, different mutations or different types of strains of this virus, like beta, delta, alpha, gamma, omicron. So we can even combine this in terms of detecting certain mutations here. Here, for example, we have like the characterization of some of these images, in which actually the good thing for us was trying to develop something that it won't be robust. Even you can have like the presence of the different strains of the virus, right? So that's something that we try to demonstrate it here. So then the next step for us was actually to implement as a high throughput assay, and that for this we basically couple or device with liquid handling machine, and then obviously or platform of imaging. So basically, such that everything is fully automated in terms of like handling a large number of samples that can be processed here. Then we also test, for example, in terms of cross-reactivity and interference. For example, in terms of cross-reactivity, how our assay performs in the presence of other viruses, other bacteria, such that we can still, I think, how basically actually our assay can perform. For example, this is in terms of the antigen detection, RNA detection. Actually, we determined that the assay is actually very, very specific. Then in terms of like interference of substances, we also tested with, in terms of the presence of certain endogenous substances, basically, that has to do in terms of like the way how the sample is collected. And then we can also see that for the majority of these tests, actually, our assay also performs well. Then our next step for us was this clinical validation. And what I want to point out here is the type of measurement that we have is dual, basically, because we can measure basically antigen and RNA simultaneously in each one of these individual particles. And we did this for saliva, and then we did this for nasal swab, and which we had actually very high specificity. Here's like the table in which we can have when we have these combined measurements, actually, we can have a very high specificity for the different types of measurements that we have. So we can also present this in terms of AUC curves to show that actually the types of measurements that we can have are very specific. So combining these two measurements, actually antigen and RNA, we can have a very specific assay in terms of determining the presence of these virus signatures in these different subpopulations of particles. So then the other interesting thing that we started to explore based on the sensitivity is if we can actually track these traces of the virus now at a much more longer periods of time. And that's what we did here for a small cohort of patients, in which this was, by the way, done in plasma, in which we can see that actually we can still have these viral signatures over a very large period of time, in terms of over 150, 200 days, in terms of like this still these EVs are carrying some of this viral content. So this we're trying to repurpose this in terms of like the long-term COVID. So that's kind of like we are very interested in this direction. So these are kind of like the main highlights of this project. So I don't want to take all the time for this, but basically as a summary, I just show you that we had adapted our single EV approach for basically the characterization of protein and RNA in viruses. We have shown that we can analyze this cargo diversity starting from a complex biofluid. So we can also show that we can track these virus signatures in EVs over a longer period of time. And we think that this type of approach we can actually be used to develop a new type of disease diagnostic tools based on single EV particles. And this was a large effort from my group. And, this is all the people involved in this project you just actually finished it this year. And with that, I will just go with your questions. Thank you very much.
Well, thank you, Eduardo. And, I know that I believe that Kim is here now as well. So, but I think, yeah, we're going to wait for the questions to the end of the next presentation too. So, well, when we have a virus particle or when we have an EV, one of the questions that comes up is, well, how sensitive can we get? Do we have to have this very long viral RNA where we can set down a bunch of probes, or can we get even more sensitive and have just a shorter RNA? And this is a place where I think that Nils Walter's lab, and we're going to hear from Alex Johnson-Buck about some of the work that they've been doing. So here we're talking about those single molecules. How sensitive can we get? How specific can we get in understanding the small things as well? So Alex, thank you so much for being here. And thanks to all members of the Walter Lab who are also on the call and can join in the discussion. So please go ahead and tell us about some of your exciting work.
Yeah, thank you all for being here. So as Ken noted, I'm Alex Johnson-Buck from the Nils Walter group at the University of Michigan, and I'm here to talk to you not about EVs in particular, but about a single molecule detection technique that we think could pair very well with EV-based analysis, EV analysis, based on single molecule kinetic fingerprinting, which is a technique that we've developed over the past several years. So, and our motivation for developing this technique is not that detecting single molecules is particularly difficult. In 2024, it's not really that challenging to detect single fluorescently labeled probe molecules. What is challenging is to tell exactly what those probe molecules are bound to with extremely high confidence or accuracy. So if we consider an assay, say a surface-based assay, where we have a target immobilized onto a surface. This could be done via a variety of ways, capture probes, it could be on an EV or anything else. Our detection probe will bind to that target, but it will also bind potentially to other species in the mixture. If this is a nucleic acid, as we've shown here, it could be a single nucleotide variants that it would also bind to some extent. That's kind of unavoidable. And it could also be binding to just the surface itself or to other sort of matrix components that have settled onto the surface. And so if we look at for instance, turf microscopy image, we can easily detect those single fluorescently labeled probe molecules. The problem is we can't tell, is this really binding to the target? Is it binding to something that's closely related? Or is it just binding to the surface itself? And both of those types of background binding occur quite commonly with both nucleic acids and especially with proteins. Both of them are somewhat sticky towards surfaces. So how do we get around this? Our solution is to actually use an unconventional approach that exploits dynamic probes rather than tightly binding probes. And these dynamic probes, of course, are a little bit lower in affinity, but they allow us to allow us to analyze their binding and dissociation kinetics so that we can tell the difference between binding to the target or to the non-target or to background, such as the assay surface. We do this by essentially analyzing the fluorescence intensity versus time pattern, which denotes binding and dissociation repeatedly to the same single molecule of the target molecule. Of the target species. And if we look at that compared to a non-target, we can often see that there's a very significant difference in the kinetics that becomes more and more apparent as you have more and more of these independent measurements, essentially, different independent binding events. And background binding is typically much more stable, and so we just see maybe it binds and then photo bleaches eventually. So what we get are video microscopy data sets that look like this, and we can see that these spots are blinking, some of them very fast, some of them rather slowly, some of them not at all. And we can analyze the pattern of the kinetics to determine this is the molecule we're looking for, or no, this is garbage, we should ignore it. And so that's not possible to do with stably binding probes in general, but with these kinetic probes, we can tell that very, very accurately and precisely. And so if we look at something like the number of binding and dissociation events that we observe per measurement window, which we call NB plus D. This is just one metric that we can use. There's sort of a lot of rich information in here, but NB plus D will help us distinguish between the true target binding and the non-target binding. As we get more and more events, we get better and better separation between the target distribution and any background binding that we observe, and we can essentially do some thresholding to get basically zero background. So we've shown that this works with microRNA. That was sort of our first application, and one of the motivations for that is just that you can't fit a whole lot of probes on microRNAs, as Ken was alluding to. And mRNA, you can load it with a bunch of different FISH probes and get a lot of fluorescence intensity from that, and some additional specificity, at least if you're not looking for a very specific mutation. But for microRNAs, you just don't have that luxury. There are 21, 22 nucleotides long typically. And so in this case, we can just capture and directly images from crude cell extracts or from serum. Because we're not using PCR, we don't have to worry about PCR inhibitors or things like that. We can just capture directly with a little bit of treatment with SDS and proteinase K to make sure that the microRNAs are liberated. We find that the short probes allow us to assay these microRNAs with low femtomolar LOD, and we don't have to use any adapter ligation or anything like that, like something like small RNA-seq would have to do, and some larger probe-based methods would have to do, which reduces some of the bias that would result from biased ligation. So that's old work, so I don't want to talk about it too much, but I just wanted to give a survey of the variety of targets that we've been able to assay. So RNA is one. We've also looked at DNA. Of course, we have to denature it in that case and then capture it on the surface. But one of the striking features of this work was that we were able to demonstrate extremely specific detection of mutant DNA. So this was, for instance, a EGFR T790M mutation, which is a single C to T mutation, which is very challenging to detect with high specificity, because if you're using a PCR-based method, often you have cytosine deamination, which can be confused for the mutation. So if we essentially had to be very careful not to expose the sample to any heat in order to introduce any of these mutations, because our technique was that specific. But we were able to detect less than 1 in 1 million mutant molecules against a large background of wild type molecules in this work, which is higher specificity than ddPCR assays available. And perhaps even most excitingly, protein doesn't allow for PCR or any analog of PCR. So if we're looking at protein analytes, it's especially important to have very specific and sensitive single molecule detection if we want to get ultra sensitive assays. And so using detection fab fragments, we were able to detect a variety of protein targets, and I think over the past few years we've extended this to 20 different protein targets, so it seems to be quite generalizable. Whenever we have tried a selection based on phage display to generate these detection fabs, I think because they don't have to be so high in affinity, it's pretty easy to find detection fabs that work really well for this technique. So it doesn't require a high affinity detection probe, and yet we're still able to get much more sensitive, much lower LODs than with ELISA, because we can reject essentially this background binding. So looking at some work that we've done a little bit more recently, so Pavel, a postdoc in Nils's lab, has been working on methods of multiplexing SIMREPs, because you may have noticed that the results that I've shown so far have just involved single-plex, single-target measurements, and we often want to measure multiple RNAs or RNAs and proteins. And of course, this technique is nice because you can achieve the same really high performance with a variety of different classes of targets. And of course, we can do something like DNA paint or exchange paint, where you have one probe introduced, remove it, introduce another probe, remove it. But that takes a lot of time, and we're already taking videos, so we want to sort of make this as efficient as possible. Of course, color-based multiplexing is really standard, so we're doing that. But also on top of that, we're layering in a kinetic multiplexing, which Pablo has been able to show. Actually, here we're showing only 6 targets. I think he can done it maybe with up to 9 targets, distinguishing 3 different types of kinetics. But on the x-axis here, we can see this is the tau on, or the bound state lifetime essentially of the probe. So we have fast dissociating, slow dissociating, and 3 different colors. And using this to distinguish, we can distinguish 6 different microRNAs and achieve low femtomolar LODs for all of them simultaneously. He's shown that this works with total RNA extracts from different human tissues, as well as different cell lines, and sort of compared these to values from the literature, and shown that this that his results match up well. Also using C. elegans miR39 as a sort of non-endogenous spike in for normalization. And he's shown that in prostate cancer patient samples, miR141 and miR375 are overexpressed in total RNA extract from the plasma or serum from these patient samples compared to those with a negative biopsy or healthy patients. Whereas miR16 is not upregulated and not overexpressed in these samples. And beyond looking at multiple microRNAs at once, he's shown that this kinetic and sort of chromatic kinetic multiplexing, as he calls it, can be applied to detecting microRNAs and proteins in the same assay simultaneously using a variety of different probes, ranging from oligos to aptamers to detection fabs. And what we can see is, just kind of to demonstrate this principle, he's looked at healthy serum versus serum from patients who had undergone CAR T cell therapy and experienced cytokine release syndrome as a side effect of that. And in CRS, one of the side effects is, or one of the biomarkers that is increased markedly is IL-6. So he does see an increase in the IL-6 in these patients. But also because of the type of B-cell lymphoma that they have, there's an increase in miR21 in their serum as well. And I'm running out of time, so I don't want to go too much longer, but I just wanted to highlight a couple of additional applications. We've sort of undertaken some forays into DNA methylation analysis and profiling recently, and this is primarily the work of Luhan Dai, a recent PhD graduate, and he's shown both with and without bisulfite treatment that we can use this kinetic fingerprinting to detect DNA methylation. I think the most exciting of these personally is probably the bisulfite-free profiling, because bisulfite treatment is pretty harsh and it often results in quite a bit of degradation of DNA. But it's required if you're going to PCR amplify the DNA and still want to detect the original methylation state for epigenetic analyses. So here we can bypass that bisulfite step entirely by just using a fluorescently labeled methylation binding domain and detect symmetrically methylated DNA or hemimethylated DNA and distinguish those from either unmethylated or sort of no DNA controls on the basis of the kinetics and the number of binding events that are observed. And we can just sort of see the different profiles for symmetric and hemimethylated here, for instance. The bound state lifetime, for instance, is quite different for symmetric versus hemimethylated DNA targets, which is pretty exciting. Maybe we can ultimately move towards a bisulfite-free workflow for detecting single molecules of methylated DNA. And Tanmai, this is the last application I'm going to talk about. Tanmai in the lab has applied this to profiling the interaction between messenger RNA mimics and microRNA that is bound to the micro-risk complex that is basically bound to AGO2 and pulled down from cell extracts onto a surface via capture antibody against AGO2. And then using a variety of probes of different lengths that mimic the different segments of the messenger RNA target that the microRNA binds to, he's shown a lot of really interesting things, which I don't have time to go into right now. But for instance, he's shown that binding to the seed sequence is dramatically accelerated by binding to the by the presence of the micro-risk complex versus the microRNA alone, which makes sense because the seed sequence is has to bind pretty quickly for efficient and rapid target recognition. There's also this sort of marked drop as you extend the target length to 9 nucleotides or 10 nucleotides. It kind of comes back up, but also the dissociation increases. So you essentially get this loss of affinity as you start base pairing with this central region of the microRNA, which makes sense because there's probably some steric hindrance there, but also this is in line with typical base pairing patterns of microRNAs. And then as you get to base pairing with the supplementary non-seed regions, you start to see increases in affinity again. And one of the more interesting things he's observed is that for most of these micro-risk complexes, you actually do see some binding to the non-seed sequence as well. So if he designs a probe against the non-seed sequence, I think these sort of orange and blue distributions of spots represent micro-risk complexes to which that non-seed sequence binds. And however, the majority is still seed binding. But you actually see a sort of a switching between seed accessibility and non-seed accessibility in a lot of these individual complexes, which is something that you just wouldn't see without this single molecule level observation. And he thinks that this is due to, I think, differential interaction of this 3' non-seed part of the microRNA with the PAS domain of AGO2. So this is sort of a small subset of the recent advances. We've been able to detect messenger RNA as well, achieved higher sensitivity, faster data acquisition, down to as little as 1 to 10 seconds per field of view, which is quite a bit lower than the original 10 minutes. We were able to use super resolution data analysis to increase our dynamic range, essentially packing more analytes into a small area and detecting them still. And for EV analysis, we're definitely more EV curious and definitely not EV experts ourselves, but we're very interested in application to EV analysis because we think it could be a really good fit for situations, particularly where you want to detect rare mutations or low abundance targets. Maybe if there's only 1 or 0 copies per EV, this could be a very valuable approach. Or if you have just a single nucleotide variant that you're looking to detect with very, very high specificity. Of course, we can assay a variety of different targets and multiplex and do super resolution imaging if we want to do some spatially resolved analysis of these EVs. This is just showing one example of some super resolution analysis, not of EVs, but of lipid nanoparticles that were loaded with therapeutic messenger RNAs that we've done some paint imaging of. And done some essentially diameter profiling of. So that I'd just like to thank all of the people that performed and contributed to this work and our funding sources, and thank you all for your attention.
Well, thank you very much, Alex, and thanks to Eduardo as well. Eduardo may have had to run to teach at this point, but I would like to allow everybody to unmute themselves and to start their videos if they would like to. Just a reminder, put your questions in the chat box, and then I will call on you as I get to those questions. And I would like to start out, Alex, with so I think there are a lot of questions that pose themselves to me. One of them is about the process of coming up with the right reagents. So you touched on that, but I would imagine that it's very important to have exactly the right affinity for those reagents to be able to observe, to really be able to use the technique correctly. So can you walk us through a little bit more about what you do? Because I assume I can't just order an antibody online. You got to come up with just the right reagents.
Yeah, so for nucleic acid, it's relatively straightforward these days. I mean, you can do in silico probe design. Often, it's helpful to empirically test like 1 or 2 candidates or something like that. And it's fairly easy to dial in the kinetics you need. For protein targets, it's true that the affinity that is ideal for our SIMREPs or kinetic fingerprinting is not perfectly aligned with what a commercial antibody would typically be selected for, right? Typically, they are selected for high affinity, slow off rate. So yeah, in general, we found that it's most efficient in terms of time, at least, to actually just select using phage display. So we've essentially ordered these from a company that does phage display selection of Fab fragments. On the other hand, we have also had some success sourcing aptamers from the literature, and with those, there's to some extent an ability to do some rational design of fast dissociating probes. So Tan Mai has published a paper on this, I think, a couple of years ago, 2022, where I think we developed an IL-8 assay and was a VEGF assay as well, using aptamers sourced from the literature. And for the IL-8 assay, I think basically we didn't have to modify the aptamer at all. Often aptamers are a little bit lower affinity than antibodies anyway, so that's kind of working in your favor. Of course, there's fewer of them available as well. And then for in the case of the VEGF, it was I think performance was optimal if we sort of made some modifications, I think, in the conserved binding region, just like maybe a single mutation resulted in much faster off rate. but still specific enough binding to be useful. So yeah, so for proteins, some additional work may be required. You could also probably screen commercial antibodies as well, but probably many of those you would have to adjust the imaging conditions if you wanted to get really fast off rates, or you just wait a while for many binding events to occur.
So what about the case where I have some EVs, I want to understand, I want to use your technique to look at a protein and an RNA. What information can we extract about co-localization as opposed to simply being there?
Yeah, so like I said, we have there's no reason you can't do super resolution analysis. So I mean, even without super resolution analysis, you can these are spatially resolved measurements, right? So if you, for instance, want to use Nile red to sort of identify and to some extent size the EVs, you can do that as a first step, and then probably add your probes for the protein and or RNA, perhaps simultaneously if you use kinetic multiplexing, or you could add them sort of in sequence. You could, if your only option was a tightly binding antibody, for instance, you had a good antibody that you want to use that was commercial, you could still do like the kinetic fingerprinting for your really sensitive, like your specific measurement of your nucleic acid, and then even add your protein probe after that, essentially, because you can flush out these transiently binding probes relatively easily, and you don't have to worry about them sticking around. So it doesn't even have to be like a bunch of different colors necessarily, as long as your EVs are obviously stably bound to the surface.
Great, great, excellent. All right. Well, I would like to go to the chat box. I know there are a few people who have some questions here. So when I call on you, please be prepared to unmute if possible, and to ask your question. So let's start with M. Cortez. I believe this question is for Alex.
Yes, I was wondering when you were explaining how the receptors are interacting with the proteins, if this is particular to the protein or particular to the sequence of the oligo, for instance, and if that is going to make it like a little bit harder to distinguish what is like the real molecule, the real ligand or antisense from any other background. That was my question.
Okay, so if I understand your question, you're asking if the sort of that on and off sort of blinking is specific to the
Like
the ligand or to the probe and target pair.
Yes, yes. If each time that we are actually targeting a particular protein is going to have like this particular flicking, and if we go and change the target, that is going to be a different one. And the same thing with the oligos, and in the oligos, how you deal with the polymorphism.
Okay, yeah, so that's, I think, maybe two separate questions. So the what we call, what you're calling the flickering frequency is really, the binding and dissociation of the probe to and from the target. And that you can think of as maybe each binding event as maybe an independent affinity measurement. So you get, I mean, it's more than that because you get kinetics as well. But you essentially get these sort of repeated affinity measurements to the same single molecule target over and over again. And so that gives us a lot more information than you would with just a stably binding probe, because you don't really know what that's bound to. I mean, maybe it's most likely that it's bound to the target, but it could just be stuck non-specifically to the EV surface or to your assay surface nearby. So I think that repeated binding is actually an advantage, and we've shown that it's an advantage. We can detect single nucleotide variants with very, very high specificity, just because the probe will exhibit different kinetics if it's binding to something even slightly different. This could be a liability potentially, as you're talking about with polymorphisms. I mean, if you want to sort of make your assay robust to polymorphisms, I guess if they're known in advance, you can design your probe against or around that, right? You could maybe have a universal nucleobase in there, or just avoid that polymorphism containing region, or you can design a series of probes. You could have more than one probe present at the same time that are designed against different polymorphisms, different single nucleotide variants.
Thank you. Thank you. Very good. There you go.
And we also, I should say, we have other variations where, I mean, you have a stably bound probe and there's a transiently binding probe that undergo fret with one another. So you get not only some added specificity there, but potentially, that stably binding probe could be designed around the region with a that has polymorphisms present that you don't want to detect for some reason, or you want to be robust to.
Thank you.
Great. Thanks for the question. Next is Ming Lin. Ming Lin, you have a question about the RNA source.
Yes. Great work first. Have you compared the RNA extracted from the plasma versus from the serum? We during the sampling, when we draw blood to get the serum, a lot of blood cells, was involved in clot formation. Then the DNA or RNA extracted from the serum and the plasma quite likely are different.
Yeah, so I assume that was question was for me, right?
Yes.
Okay. Yeah. Thanks. Yeah. Thanks for the question. So I think the short answer is no. I don't think that we've systematically tested the differences between serum and plasma. I think Pavel may be in a better position to answer this. Like if he's done any comparisons between serum and plasma recently. Pavel, you can feel free to enter your response in the chat if you have an answer to that. But I think generally speaking, we've mostly used serum, and it's generally been serum that's collected sort of by our collaborating labs in a very specific way to preserve RNA, and to I think there's a high concentration of EDTA in there, among other things, to really make sure that the mRNA is as stable as possible throughout the collection process, and then sort of rigorously stored and frozen and handled prior to it coming into our hands. Because there can be quite big differences from protocol to protocol as well in what kind of microRNA results you get. But
Yes.
I think
Okay.
we don't have as much experience with plasma. Actually, I think there's maybe been a little bit. We have done a few plasma samples here and there with microRNA.
Okay. Good. Thank you.
Yeah, Pavel, did you want to add anything to that? If so, just you can feel free to speak up. But if not, we have a couple of questions here from David Juncker. Please go ahead.
Okay, no, I just add in the chat also, like we also check from the same question, like we generally get a bit less count when we use the RNA extract from serum compared to the RNA extract from plasma. Well, that's why we always use like RNA extract from a single source, and that is serum, which is available for us in this particular experiment case.
Great. Okay. Thank you.
Thanks, Pavel.
Helpful. David, hi. Nice to see you.
Hi, Ken. Great to see you. Thanks for reviving this, and thanks for the great talks. So I from your question, I was just wondering, I mean, we have a bit of complementary here with the padlock probes, which kind of open up inside of EVs, and your probes, so which kind of bind on and off. So is there a way that your system would make sense of delivering inside of EVs, or would that give a benefit? I mean, I'm kind of speculating here, and could that address maybe some of the challenges of actually inserting probes inside of EVs? Because I think that's, I mean, obviously there's some solutions, but I think for small EVs, at least from what we're seeing, this can be quite tricky.
Yeah. So I mean, we're very interested in this. It's not something that we've really, we've been developing the sort of fundamental technique itself, not so much applying it to EVs at this point. But I think the fact that the probes, certainly for nucleic acids, can be quite small is probably advantageous in terms of getting them into EVs without having to sort of blast them apart and make them, so permeable that the contents leave. Yeah. So what was the other part of your question that I'm missing? So in terms of, well, I think if we get them into EVs, so there's sort of the question of in general, we have this sea of probes, right? That's just diffusing freely in solution above the sample, and we basically reject their fluorescence by using turf, or high-low or other techniques like that. If we get a probe inside of an EV, if we want it to stay in there, I mean, we actually do have variations of this technique where there's sort of intramolecular fret, but there's still that transiently binding signal. So you have binding of two adjacent sequences, and depending on whether that second sequence is bound, you get fret or no fret. So that's another option if we wanted to essentially get a probe into an EV and just keep it there, and just have this blinking signal that still gives us very high specificity to that adjacent sequence there, if you wanted to detect a mutation, for instance.
Okay, so yeah, well, because inside of an EV it would be very confined, right? So it would,
Right.
you would expect that it would change the kinetics quite substantially of your blinking.
Yeah,
Potentially, yeah,
I
it's
think this is a
the
really
on
key
rate.
question.
Yeah.
Yeah,
So,
I would like, I'm wondering, Chook, are you still there? I'd like to invite you to maybe give some of your experience with the permeabilizations that you had to do for your assays, what worked, what didn't, things that maybe you haven't tried and want to try. I also see Zach and Ahmed on here. I know that both of you have done some work with permeabilization, probably other folks too on the call. So I'd like to just hear a little bit more about what everyone's experience has been with this.
Yes. Hi. Hi, everybody. My name is Drew. I'm sorry, my computer has some problems, so it's in with the IT department. So I need to log in with my friend's computer, and I cannot change the name. I'm sorry. I'm not that tech.
She's the first author on the paper that Eduardo presented. Yeah.
Yes,
Okay.
yes. So yeah, my name is Drew. So yeah, to share a little bit of the experience with the permeabilizations process. So we have done in our particular projects, we have tried with two methods of permeabilizations for the EVs and the virus particle. So we have tried with the commercialization kits, the eBioscience, which has the ability to have the formaldehyde to kind of like fix the EV's surface, and then we permeabilize that one as if it's working with the cell's membrane, and then we deliver our probe, which is the molecular beacons, in for it to hybridize inside the EVs. That one work well in our detections. However, what we have seen is that the EVs, when it get permeabilized with that harsh conditions, we need to keep it on the ice for a long time, and that happen to have release some of the RNAs within the EV structure. So when we do it, that one we observe a little bit of the decrease in the signal, though still we can detect the signal within some EVs still. The second step the second method that we was using is publicized in the virus paper, which we use the TE buffer. TE buffer is the TRIS EDTA buffer, which we have been using it to stabilize our probe, which is the molecular beacons. So it happened to help also is make the membrane, the bilipid layers membranes, to be more dynamic, so that it can help when we incubate the sample in a elevated temperature. Typically, we use as 37°C for 2 hours. Is make the membranes become more dynamic, and it's allow the molecular beacons, small nucleotide sequence, to get inside the EVs and help to hybridize, break open the molecular beacons in its pristine conditions, and then hybridize with the RNAs of the virus or the EVs that we have tried. So we observe with these methods is getting a little bit better signal compared to the previous methods, where we have need to permeabilize, and like kind of like you can imagine that we need to poke a permanent hole on the surface of the membrane. So it's kind of like we think that the T buffer is working as beautiful as the way like it make the membrane just enough dynamics that is allow things to go inside. And once we move the sample from the incubations, bring it back to a lower temperature, it lower the dynamic of the membrane, so it's kind of like seal off, and the probe stays inside the EVs. So for these methods, we observe a very good and consistent result when we repeat the measurements with our like spike sample. And also for the we have tried with the last experiment with the long COVID detections for plasma sample. It's worked perfectly good with the plasma sample. I have repeat some of the patient sample for a duplicate different days that I was doing. It's still giving the correct trend and signals. So it's very stable from what we have observed.
Can I follow up?
Yeah.
Yeah, I have a question actually. That's great. Thanks so much. So for the permeabilization, we have been working with on that too. So it seems to us that big EVs are kind of fairly easier to permeabilize than small ones. So have you
Yes.
tried to check like with maybe a membrane dye or just by comparing the fluorescence whether you see like more or less permeabilization? I mean, I guess it's a bit hard, but also whether you lose also then the RNA inside of the EV or not. I don't know if you spike it in with a fluorescent label, for example. I don't know if you have any comments on that or any
Yes.
data.
Yeah, that's very good questions also. So for our assay is a multi-parametric assay. So we do detect the RNA and the surface proteins as well. For our assay, we see the loss, like I have mentioned just now. So we see a loss a little bit in the signal when we do with the commercialized kit. But for the proteins on the surface, we do detect with the antibody fluorescence antibody. So for that one, we don't see much of the signal, probably because the fluorescence from the antibody on the surface is much brighter compared to the signal also from the small molecular beacons that is got inside of the EVs. But we do see when we capture the EVs on the surface and permeabilize by the eBioscience kits, it do reduce the amount of the EVs that was captured on the surface as well. So I think it's kind of somehow it's a little bit disrupt, or the proteins or the antigens on the surface is got lose a little bit during that process as well.
Okay, thanks.
Alex, and for the LNP work that you showed, what did you do to expose there?
Yeah, so I mean, for the LNP work, it's mostly been a question of just, can we do super resolution imaging of the particles themselves? So it's not permeabilizing them necessarily. I mean, I think Tanmay has done a little bit of work to basically use I don't remember what detergent you use. Maybe Tanmay, you could comment. But kind of a mild detergent to sort of permeabilize them. He's been able to detect the messenger RNA inside of them using just ribogreen. I think it's sort of like the stage at which we're at right now. Not necessarily introducing specific probes in yet.
Got it.
So it's Triton X100.
Triton X100, thanks. Yeah.
Good.
Yeah, so at a relatively low concentration, I think.
Yeah, it's very low concentration. Yeah.
And these are, quite different particles from EVs. So I'm sure that the same approaches here won't necessarily generalize to EVs. These are quite cationic lipids, as I understand it.
So sorry, what was that? Triton X100? Is that what you were using?
Yeah.
Yes. It's Triton X100 at 0.01% volume by volume. It's sufficient to permeabilize enough to introduce the ribogreen dye inside and to image the RNA inside. Yeah.
And yet we can still detect the I think we have some evidence that the RNA is, it hasn't leaked out essentially, right?
Right.
I guess if it had, then we wouldn't be detecting it at the surface along with the EVs or with the LNPs.
Yeah.
Well, certainly a lot to to figure out, optimize as we try to get different sorts of affinity reagents into different sizes of EVs with different compositions. So lots of work still to do. But David, I think you had one more question, didn't you?
Yeah, well, I'm actually I had also I'm happy to the other group is also there, but yeah, I asked my question. But maybe actually coming back to your super resolution approach, could you tell us a bit about the trade-off between sensitivity and time, or basically the time scales that we're talking about? I mean, I know we're also doing assays, and you said like both for proteins and maybe DNA 0.9998%. That means you need to measure lots of events before you can have these low percentages. So what's your field of view? What's the density, and how long does it take to get these kind of numbers, maybe for RNA and also for proteins actually?
Yeah, so I didn't have a chance to go into this in much detail, but on the last slide, I mentioned that we've made efforts to really reduce our acquisition time. So we have a number of new approaches. One of them is published already. Is one using sort of intramolecular FRET probes. I kind of alluded to those as we were talking about ways of sort of getting probes inside of EVs. That might be an interesting approach. But that, I think, we've been able to detect in about as little as 10 seconds per field of view, and this is maybe a single field of view of a under 60x objective. So that's maybe a 200 micron diameter at best, at largest. So I don't know, maybe you're talking about thousands of EVs, a few thousand EVs at most in that field of view. So obviously, if you're going to try to image 1 million EVs, that's going to take quite some time. Probably we're, in most cases, not going to be that's going to be the limiting factor in the lowest abundance of a mutation we would be able to detect is the imaging throughput rather than the specificity, which is really not a limiting factor at this point.
Oh.
But we also have some other newer approaches that I'm not really ready to talk about yet that basically, allow us to even get that down to, 1 or 2 seconds, which I think is, that could be a game changer as far as throughput.
Yeah. Okay. Well, thanks so much.
Thanks for all the insightful
any
questions.
last words from anybody here? Because we're getting to the end of the hour, and I think we've had two really impressive presentations. We heard about how we can use detect different kinds of molecules in EVs and viruses, and how that might be applied to clinical situations. And then we've heard about a really nice cutting edge way of understanding the presence and also how much of a particular molecule is around in a particular sample. So I hope to see more and more crosstalk like this. So the people who are developing great new methods can talk with the EV people on this call, and we can all come up with new ways of learning about what the EVs are trying to tell us. So thanks so much to Eduardo, to Truk, who also joined and helped with the questions, and thanks to you, Alex, for presenting, and to other members of the Walter Lab who were here. So please keep up the great work, and we hope to see you all again on an EVClub sometime soon. So thanks, everybody. Have a great rest of the week, and bye for now.
Thanks so much.